In this video, we are going to once again learn about enzymes in detail, many important topics related with enzymes. A living system is a curious biochemical factory. It is an open, steady state system in which biochemical reactions take place for the maintenance of life process. These reactions are mediated by biological catalysts called enzymes, and many of them are controlled and coordinated by hormones and vitamins. Enzymes selectively accelerate the rate of reactions, maintain their orderliness, and ensure that they take place in an appropriate cellular sites. As a rule, all biochemical reactions have to pass through an activated complex whose formation may take great deal of energy. However, living cells can carry out biochemical reactions at appreciable rates at mild temperatures, low pressure, and pH volumes close to neutrality. Once again, let's learn this. A living system is a curious biochemical factory. It is an open, steady state system in which incessant biochemical reactions take place for the maintenance of life processes. These reactions are mediated by biological catalysts or enzymes, and many of them are controlled and coordinated by hormones and vitamins. Enzymes selectively accelerate the rate of reactions, maintain their orderliness, and ensure that they take place in the appropriate cellular sites. As a rule, all biochemical reactions have to pass through an activated complex whose formation may take a great deal of energy. However, living cells can carry out biochemical reactions at appreciable rates at mild temperatures, low pressure, and pH values close to neutrality. This is certainly due to the activity of enzymes, which can, on one hand, accelerate metabolic reactions and, on the other hand, regulate them. These enzymes can speed up the otherwise slow organic reactions and control their rate. Enzymes are exceedingly efficient catalytic biomolecules with controllable activity. They serve as regulators of metabolism, selectively accelerate the approach of biochemical reactions to equilibrium, and increase the efficiency of energy transactions. Uncatalyzed bioreactions are slow and sluggish, and enzymes can accelerate them and control their rate. The substances upon which enzymes act are called substrates. A living system may contain many thousand kinds of enzymes positioned in definite locations. Many of them do not act independently, but in active teams or chains with mutual control and coordination. Such enzyme chains are called multi-enzyme systems. They bring about chains of interrelated and sequential reactions known as metabolic pathways. In such a pathway, the product of one reaction forms the substrate of the next reaction. Some enzymes exist as active functional complexes called multi-enzyme complexes to catalyze multi-step reactions or reaction complexes. Pyruvic acid dehydrogenase complex mediates the conversion of pyruvic acid to acetyl-CoA. Fatty acid synthetase complex mediates the conversion of acetyl-CoA and malonyl-CoA to palmitic acid, etc. are examples. Control on enzymes. Enzymes are subject to a number of biological controls. For example, the rate of their synthesis and final conversion are under genetic control and are often influenced by the concentration of their substrate and the products of their reactions. The biosynthesis of enzymes itself is enzyme catalyzed. Coenzymes and endoenzymes. All enzymes are produced within the cell. Still, in multicellular organisms, some of them act outside the cell. They are called exoenzymes or extracellular enzymes. Those which function inside the cell are called endoenzymes or intracellular enzymes. Hydrolytic digestive enzymes. Hydrolytic digestive enzymes of the alimentary canal are exozymes. And hydrolytic digestive enzymes of the alimentary canal are exoenzymes like amylase, protease, lipases, where all metabolic enzymes and respiratory enzymes are endoenzymes. Cytochromes. For example, cytochromes are endoenzymes. Constructive and inducible enzymes. Some enzymes are present always as normal cellular constitutive and inducible enzymes. Some enzymes are present always as normal cellular constituents. They are called constitutive enzymes or housekeeping enzymes. For example, cytochrome oxidase. Others are produced only when their substrates are available. In other words, the presence of the substance induces their production. So they are called inducible enzymes. A typical example is lactate dehydrogenase. Its synthesis is induced by the presence of lactose. Cymases and cymogens. Most enzymes are secreted as active forms called cymases or active enzymes. So they can act directly upon their substances without undergoing any chemical modification or functional maturation. All intracellular enzymes are cymases, glycolytic enzymes, Krebs cycle enzymes, etc. On the other hand, some enzymes are secreted in their inactive or precursor forms called cymogens or proenzymes. They are made actively functional by certain activating enzymes or agents. The extracellular digestive enzyme pepsin, trypsin, and chymotrypsin are typical examples. They are secreted as pepsinogen, trypsinogen, and chymotrypsinogen respectively. Pepsinogen is activated by HCl and trypsinogen by the enzyme enterokinase and chymotrypsinogen by trypsin. Extra extremozymes. Extremozymes or high temperature enzymes are the enzymes which function optimally under extreme conditions of temperature, pH, etc. Examples are the enzymes of hyperthermophilic sulfur reducing bacteria. These bacteria grow and divide at optimum rate well above 100 degrees Celsius and their enzymes are most active above 100 degrees Celsius. Next is the enzymes. Enzymes are antibody enzymes. They are catalytic antibodies which can function as enzymes to catalyze specific biochemical reactions. They are inducible enzymes produced only in response to the presence of a specific antigenic molecules. Next are the ribozymes. There are two major chemical classes of enzymes, ribozymes and proteinaceous enzymes. Ribozymes or RNA enzymes. Ribozymes or RNA enzymes are the catalytic or autocatalytic RNA molecules with sequence-specific cleavage activity. There are two major classes of enzymes, ribozymes and proteinaceous enzymes. Ribozymes or RNA enzymes are the catalytic or autocatalytic RNA molecules with sequence-specific cleavage activity. Autocatalysis is the phenomenon in which an enzyme catalyzed reaction is promoted by its product. They act as a RNA cutting enzymes and contribute to their own splicing and also to the functioning of ribosomes. They have been detected in viruses, plants, and mammals. The catalytic property of RNA was first reported by Stanley Altman, Thomas Sches, and Zug and Grabowski in 1982. For the discovery of Sches and Altman received the Nobel Prize in Chemistry in 1989. ആ ഹോർലിക്സ് ഒന്ന് മാറ്റി വയ്ക്കണേ. എന്നാൽ ശരി. The discovery that a non-protein macromolecule can serve as biological catalyst was marked a deviation from the discovery that a non-protein macromolecule can serve as biological catalyst was a marked deviation from the prevailing concept that all enzymes are proteins. Self-splicing rRNA of ribonuclease P are examples of ribozymes. There are two categories of ribozymes: self-splicing ribozymes, RNA splicing ribozymes, and self-cleaving ribozymes. The fundamental reactions with which ribozymes catalyze are transesterification and sequence-specific cleavage of hydrolysis of phosphodiester bonds, phosphorylating RNA molecules. They can also catalyze peptide bond formation. A ribozyme which can split other RNAs is called hammer-headed ribozyme because of its shape. When bonds are cleaved by ribozymes, the products will have 3-hydroxy and 5-phosphate termini, in contrast to the 5-hydroxy and 2 to 3-phosphate termini of the products of the alkaline hydrolysis of RNA. The substrates of ribozymes are mostly RNA molecules. The substrates of ribozymes are mostly RNA molecules. Sometimes the RNA substrate may form a part of the ribozyme. Ribozymes and their RNA substrates often enter into base pairing interactions to bring the substrate into reaction. It is strongly held that many of these spliceosomes are ribozymes evolved from primordial self-splicing RNAs. Spliceosomes are multi-component ribonucleoprotein complexes involved in RNA splicing, removal of introns from nuclear pre-mRNAs. The current concept about ribozymes is that they are the vestigial remnants of primordial living systems. According to this concept, in the precellular primordial biosphere, nucleic acids functioned as the first enzymes of the living system, and in the course of evolution, proteins replaced them as enzymes. The current concept about ribozymes is that they are the vestigial remnants of primordial living system. According to this concept, in the precellular primordial biosphere, nucleic acids functions as the first enzymes of the living system, and in the course of evolution, proteins replaced them as enzymes. Next is the proteinaceous enzymes. Proteinaceous enzymes are electrically charged, high molecular weight catalytic proteins which have controllable activity. Their catalytic property depends upon their intact organization. If an enzyme is denatured, the catalytic activity depends upon their intact organization. If an enzyme is denatured, associated or dissociated into subunits or degraded, broken down to amino acid units, the terms denatured is different from and degraded is different. When we say an enzyme is denatured, the actual meaning is that they dissociated into subunits. And when we say the enzyme got degraded, the they are broken down to amino acid units. Its catalytic activity usually gets lost. So, denatured means dissociated into subunits. Degraded means broken down to amino acid units. Its catalytic activity usually gets lost. The molecular weight of enzymes ranges from 12,000 to over 1 million. What are the properties of enzymes? Enzymes are specialized proteins which serve as exceptionally versatile catalysts in biochemical reactions. Some of their special properties are the following. Enzymes are organic catalysts with controllable activity. This property enables them to change the metabolic rate in relation to the environmental conditions. Enzymes are organic catalysts with controllable activity. Enzymes are specialized proteins which serve as exceptionally versatile catalysts in biochemical reactions. Some of their special properties are the following. Enzymes are organic catalysts with controllable activity. This property enables them to change the metabolic rate in relation to the environmental conditions. Enzymes are less stable and thermobile molecules, so they will be easily inhibited, inactivated, or destroyed by high temperature extremes of pH, UV rays, heavy metal ions, etc. Enzymes are less stable and they are thermobile molecules, so they will be easily inhibited, inactivated, or destroyed by high temperature extremes of pH, UV rays, heavy metal ions, etc. Enzymes have exceedingly high catalytic efficiency or catalytic power. Enzymes are exceedingly efficient catalysts under optimal conditions. Most enzymatic reactions proceed 10 raised to 8 to 10 raised to 11 times faster than the corresponding non-enzymatic reactions. Moreover, the turnover number of the most enzymes is well above 1000. In extreme cases, it may go beyond even 1 million. Turnover number is the number of substrate molecules converted to product molecules by one enzyme molecule in 1 second. Enzymes do not alter the thermodynamic properties of the reactions they catalyze, but only alter the kinetic properties concerned with the rate of the reactions. In other words, enzymes do not push reactions uphill, nor do they shift the equilibrium point of reactions, but only increase the rate at which the equilibrium is reached. So they accelerate the onset and the velocity of reactants without altering their route and affecting their equilibrium point. Enzymes catalyze only energetically feasible reactions. Enzymes catalyze only Energetically feasible reactions. Most reactions are very slow and they do not occur at perceptible rates in the absence of enzymes. Enzyme catalyze only energetically feasible reactions. Most reactions are very slow and they do not occur at perceptible rates in the absence of enzymes. Enzymes speed them up and control their rate. Enzymes increase the rate and effectiveness of reactions by considerably lowering the free energy level of the activation barrier of reactions. Enzymes increase the rate and effectiveness of reactions by considerably lowering the free energy level of the activation barrier of reactions. In other words, enzymes increase the rate of actions by lowering the free energy of activation which a substance must acquire before it can react. So reactions can take place at normal body temperature. Enzymes catalyze the conversion of the reactant's energy to a different form with high efficiency. In photosynthesis, light energy is converted into chemical bond energy. In mitochondrial oxidative phosphorylation, chemical bond energy is transferred to energy currency of ATP. In muscular contraction, the chemical energy of ATP is converted to mechanical energy. Enzymes catalyze reactions under mild conditions. In neutral aqueous solutions at normal pressure, low temperature, and pH close to the neutral level or isoelectric point. Enzymes are neither consumed nor permanently altered, affected, or destroyed by the reactions they catalyze. In fact, enzymes do not react with the substrate and hence are regenerated at the end in their original form. So they can go through many cycles of reactions without changing the nature or properties of the end product. Enzymes are extremely specific in their action and highly selective in their choice of the substrate. In other words, they have extreme reaction specificity and substrate specificity. This is because they have high specificity with respect to the nature of the reaction and the structure of the substrate. So, a particular enzyme can catalyze either a specific reaction or a set of closely similar reactions of the same or similar substrates. This high specificity enables them to drive a metabolic process through strictly specific channels. So, enzymes are extremely specific in their action and highly selective in their choice of the substrate. Enzymes have exceptionally high effectiveness for lowering the energy level of activation barrier. Enzymes can catalyze a broad spectrum of reactions. The spectrum of enzyme catalyzed reactions is very broad. It includes hydrolysis, polymerization, isomerization, oxidation reduction, dehydration, aldol condensation, group transfer reactions, free radical reactions, and so on. Enzyme action is under biological regulation. The activity of enzymes is under the inductive or inhibitory regulation of biological factors which can stimulate or inhibit enzyme action. As a result of this regulation, substrate utilization and product formation conform to the needs of the cell or organism. Reversibility of enzymatic reactions. Most enzymatic reactions are reversible in the sense that the same enzyme can govern forward and backward reactions. However, in some cases, forward and reverse reactions are mediated by different enzymes. The synthesis and concentration of enzymes are in dynamic balance and are genetically controlled. The biosynthesis and final concentration of enzymes are genetically controlled and well within the normal limit. In fact, enzyme synthesis itself is enzyme catalyzed. Thus, all biological reactions are enzyme catalyzed and hence genetically controlled. Next, naming and classification of enzymes. Let's learn about the naming and classification of enzymes. It has been estimated that a living cell contains as many as 10 raised to 4 enzyme molecules capable of catalyzing nearly 2000 odd kinds of reactions. Naming and classification of enzymes. It has been estimated that a living cell contains as many as 10 raised to 4 enzyme molecules capable of catalyzing nearly 2000 odd kinds of reactions. Slightly more than 1800 kinds of enzymes are known to date, and about 115 of them have been isolated in crystalline form. During the early period, no systematic scheme was adopted for the classification and nomenclature of enzymes. In 1998, the French botanist scientist devised a practical scheme for naming enzymes. According to it, the trivial working nature of an enzyme can be constructed by adding the suffix "ase" to its substrate. So, according to it, a trivial working nature of an enzyme can be constructed by adding a suffix "ase" to its substrate. For example, sucrose plus "ase" sucrase. Later on, enzymes were named by using the name of the substrate, nature of the reaction, or both, and the suffix "ase." For example, urease, dehydrogenase, lactic acid dehydrogenase, etc. In 1961, the Committee of Enzyme Nomenclature adopted a new scheme, which is now accepted as the official scheme. According to it, an enzyme is given two names: trivial name and the systematic name. The former is constructed from the name of the substrate, type of the reaction, and the suffix "ase." For example, lactic acid dehydrogenation plus "ase" gives lactic acid dehydrogenase. The systematic name also is constructed from substrate, type of reaction, and the ending "ase, but in a different way. For example, lactic acid dehydrogenase is written as lactic acid NAD oxidoreductase. Lactic acid, that is the substrate, plus NAD, substrate to oxidoreduction, that is the type of reaction, along with an "ase", that is the ending. So two names are there. First one is the trivial name along with the systematic name, which are the two types: trivial name and systematic name. So first we will be writing a lactic acid substrate 1, NAD substrate 2, oxidoreduction, that is the type of reaction, is ending. Several enzymes such as pepsin, trypsin, chymotrypsin, etc. still retain the traditional names which are related either to the substrate nor to the reaction. So trivial names, for example, let's study the lactic acid dehydrogenase. Trivial name is lactic acid. Lactic acid trivial name is lactic acid dehydrogenase, alcohol dehydrogenase, glucose oxidase, hexokinase, carboxyl esterase, urease, acetyl CoA synthetase, etc. are trivial names which are created just by adding the substrate and the type of reaction along with the ending ASC. ACE. But their systematic names are like lactic acid NAD oxidoreductase, alcohol NAD oxidoreductase, beta D glucose oxygen oxidoreductase, ATP dehexose 6-phosphortransferase, carboxyl ester hydroxylase, carboxyl ester hydrolase, urea amido hydrolase, and acetate CoA ligase. Urease, we just call it as urease in the case of trivial name. But when we just write the systematic name, urea amido hydrolase, like that is the systematic name. Based on the nature of the reaction, the Commission of Enzymes has recognized 6 classes of enzymes as follows: oxidoreductases, transferases, hydrolases, lyases, and isomerases, along with ligases. Oxidoreductase. Enzymes involved in oxidation reduction reactions. They include oxidases, peroxidases, dehydrogenases, and they are holoenzymes with a coenzyme for accepting or donating hydrogen or electrons. They are holoenzymes. Then transferases are there, enzymes which catalyze group transfer reactions. Reactions which involve the transfer of chemical group from one substrate to another, like phosphoryl transferase, transaminases, transpeptidases. All those are transferases. Next are the hydrolases, enzymes involved in the hydrolytic breakdown of chemical bonds, like peptidases, glycosidases, esterases, etc. First, we were talking about the oxidoreductase. That are the enzymes involved in oxidation reduction reactions and are holoenzymes. For example, oxidases, peroxidases, dehydrogenases, etc. And they have coenzyme portions for accepting or donating hydrogen or electrons. Next is the transferases. Transferases. Enzymes which catalyze group transfer reactions. Reactions which involve the transfer of a chemical group from one substrate to another. phosphoryl transferase, transaminase, transpeptidases, etc. Hydrolysis are helping in the chemical hydrolytic breakdown of chemical bonds. Lyases are there, enzymes which remove chemical groups not by hydrolysis from substrates, leaving double bonds or add chemical groups to double bonds. And hydrases, decarboxylases, etc. So that is lyases. Isomerases are there, enzymes which catalyze the isomerization, molecular reorganization of substrates, epimerase, mutases, etc. So isomerases are there. Lyases also remove chemical groups. Okay. Isomerases enzymes which catalyze the isomerization, molecular reorganization of substrates, epimerases, mutases, etc. And ligases or synthetases, enzymes which catalyze the condensation polymerization of molecules to covalent bonding, accompanied by the cleavage of an energy bond of ATP to another triphosphate. Peptide synthetase, DNA ligases, etc. The above mentioned system of enzyme classification is often called OTHLIL system. The acronym OTHLIL is constructed by using the initial letters of the 6 classes of enzymes like OTHLIL, OTHLIL, OTHLIL. When we expand all these terms, oxidoreductase, transferase, hydrolase, lyases, isomerases, and ligases. Okay, so OTHHIL. Now let's learn about the molecular organization of enzymes. Based on molecular complexity, two kinds of enzymes can be recognized: simple enzyme and complex enzymes. Simple enzymes are simple proteins, whereas complex enzymes are conjugated proteins. Conjugated in the sense they have portions other than protein. A conjugated or complex enzyme is known as a holoenzyme. It consists of, earlier we have learned some type of enzymes like oxidoreductase are holoenzymes. That's a type of conjugated enzyme. Okay, so a conjugated or complex enzyme is known as holoenzyme. It consists of a protein part and a non-protein part, but this act together as a single unit. The protein part is known as apoenzyme and the non-protein part a cofactor. Protein part is called protein. Whole system is called holoenzyme, but the protein part is called apoenzyme, and the non-protein part cofactor. Non-protein part is a cofactor. Cofactors are indispensable for the activity of conjugated enzymes. Neither the apoenzyme nor the cofactor is active by itself. They bind together loosely or tightly and form an active apoenzyme cofactor complex known as holoenzyme, which is functional. Cofactors and coenzymes. Cofactors are either inorganic ions like Na+, K+, Mg2+, Mn2+, Fe2+, Zn2+, CO2+, etc., or non-protein organic molecules. They are inorganic ions. They can either be inorganic ions or non-protein molecules. Organic cofactors are generally called coenzymes. Examples of coenzymes CoA, CoQ, NAD, FAD, etc. are coenzymes because they are organic. In combination with their respective apoenzymes, serve as the acceptors, carriers, and donors of atoms or chemical groups. For example, NAD, FAD, CoQ, etc. serve as acceptors, carriers, and donors of hydrogen. CoA serve as the transfer of acetyl group. CoA serve as transferer of acetyl group. Some coenzymes or some metal metallic ions remain tightly and covalently bound to their apoenzyme. They are called prosthetic groups. The iron porphyrin complex, ferroporphyrin of cytochromes and hemoglobin, the iron containing part of cytochromes and hemoglobin, the flavin part of flavoproteins, etc. are examples. Cofactors confer reactivity on enzymes in two ways: by serving as a bridge between enzyme and substrate, by actively participating in the reaction. Cofactors confer reactivity on enzymes in two ways: by serving as a bridge between enzyme and substrate, by actively participating in the reaction. These are the two methods. Coenzymes are of two major types, namely those participating in redox reactions, which involve the transfer of hydrogen atom or electrons, and those taking part in reactions which involve the transfer of chemical group NAD, NADP, FAD, FMN, etc. are examples of the first category. CoA, TPP, biotin, etc. are examples of the second category. Second category, those taking part in reactions which involve the transfer of chemical group. In enzyme action, a change occurring in the substrate will be counterbalanced by an opposite change in the coenzyme. So the coenzyme serves as a co-substrate. For example, oxidation of the substrate will be counterbalanced by the simultaneous reduction of the coenzyme, as revealed by the following reaction. In case of a lactate molecule converting into pyruvate, so lactate dehydrogenase is the enzyme involved in that process. Lactate into pyruvate, and lactate dehydrogenase is present there. So it converts NAD plus to NADH plus H plus. So when this process is happening, there is something else happening into the cofactor which counterbalances the reaction. Something happens in case of substrate, and the coenzyme acts as a co-substrate by undergoing the exact counterbalancing reaction. If the reaction is reversed, the opposite effect will be produced in the substrate and the coenzyme. In some reactions, a particular group or radical is transferred from one substrate to another, or from a coenzyme to a substrate. In some reactions, a particular group or radical is transferred from one substrate to another, or from a coenzyme to a substrate. Here also the coenzyme functions as a co-substrate. Coenzyme always function as a co-substrate. Remember that. And got the example of a lactate converting into pyruvate with the help of lactate dehydrogenase, and the NAD plus is transforming into NADH plus H plus. Now let's learn about isoenzymes. Isoenzymes or isozymes are the molecular variants or polymorphic forms of an enzyme. Isoenzymes or isozymes are the molecular variants or polymorphic forms of an enzyme. In other words, they are the different molecular forms of the same enzyme. They have almost the same molecular weight and similar biological function, and they catalyze the same reaction. But they are different in molecular structure and kinetic properties. Differences in their properties and molecular organization are genetically determined. More than 100 enzymes are known to exist as isozymes. Lactic acid dehydrogenase, LDH, the enzyme which catalyzes the conversion of pyruvic acid to lactic acid, is the most extensively studied one. It is known to exist in 5 different molecular forms: hexokinase, acid phosphatase, alkaline phosphatase, nucleotide phosphatase, cholinesterase, malic acid dehydrogenase, glutamic acid dehydrogenase, glucose 6 phosphatase, phosphate dehydrogenase, creatine phosphokinase, tryptophan synthetase, aldolase, amylase, lipases, etc. are also said to be isozymes. The molecular variants of an enzyme occurring in different species of organisms are called interspecific or phylogenetic isozymes, and those occurring within the same species are called intraspecific or ontogenic isozymes. Isozymes are of two groups: homologous and analogous. Homologous isozymes have almost similar molecular structure and catalytic properties. But analogous isozymes have similar reaction properties but different molecular organization. Isozymes are of two types like intraspecific or phylogenetic isozymes, and those occurring within the same species are called intraspecific or ontogenic isozymes. Isozymes are of two groups, homologous and analogous. Homologous isozymes have almost similar molecular structure and catalytic properties, but analogous isozymes have similar reaction properties but different molecular organization. Homologous isozymes are believed to have the same genetic origin produced of the homologous isozymes are believed to have the same genetic origin, products of the same gene, whereas analogous isozymes are believed to have evolved from different ancestral genes, products of different genes that are located on different chromosomes. Some isoenzymes are there. Homologous isozymes are believed to have the same genetic origin, products of the same gene, whereas analogous isozymes are believed to be evolved from different ancestral genes, products of different genes that are located on different chromosomes. Some isoenzymes are like hexokinase, acid phosphatase, alkaline phosphatases, nucleotide phosphatase, choline esterase, malic acid dehydrogenase, glutamic acid dehydrogenase, glucose 6 phosphate dehydrogenase, creatine phosphokinase, tryptophan synthetase, aldolases, amylases, and lipases. Then there are metalloenzymes. These are the enzymes which require metallic ions for their activity. These are the enzymes which require metallic ions for their activity. They require metallic ions, so that is why they are termed as metalloenzymes. Carbonic anhydrase and carboxypeptidase require zinc. Magnesium is necessary for hexokinase, peptidases, and enolases. Manganese is essential for phosphoglucomutase and dipeptidase. Salivary amylase require chloride ions. Copper is necessary for thyrosinases. Iron is required for cytochrome oxidase, catalase, and peroxidase. Calcium activates lipases and elasthases. In some cases, the removal of the tightly bound metal will not considerably affect enzyme activity. At the same time, addition of metallic ion enhances enzyme activity. Such enzymes are called metal activated enzymes. Activation of carboxypeptidase by zinc is an example. Metal activated enzymes. Now let's learn about the functional organization of enzymes. An enzyme molecule has several functional regions, each with a specific function. A small portion of such a region serves as the specific site for binding with the substrate and also for converting the substrate to a product. So it is called active site, which we already know. Active site or active center. Another name is active center. Here, the substrate reacts with the enzyme by selective attachment, followed by breaking and making of bonds. The active site includes the peptide chains and side chains, which come in direct contact with substrate. The active center of conjugated enzymes include cofactors also. In an oligomeric conjugated enzyme with at least two identical subunits, different subunits may share the same active site. Active site has no rigid shape since its amino acid residues can realign themselves for substrate binding. An active site is said to have two portions, namely contact site, substrate binding site or anchor site, and the catalytic site or conversion site, catalytic site. Catalytic site and anchor site. Anchor site will be there and catalytic site will be there, or contact site will be there. Contact site will be there and catalytic site will be there. Contact site and catalytic site will be there. Okay. The binding of the substrate occurs at the contact site and its catalytic conversion to the product occurs at the catalytic site. The concept of this differentiation now appears to be arbitrary or doubtful, but still we are learning that. So, functional organization of enzyme. A simple enzyme is there. The catalytic site will be there, contact site will be there, and the active site will be there. So, in a holoenzyme, cofactor will be there, and catalytic site will be there, contact site will be there, and apoenzyme will be there. So, that is the structure of a holoenzyme. But when we talk about allosteric enzyme, allosteric site will be there, contact site will be there, and active site will be there, and a catalytic site will be there. So there is a specific portion called allosteric site that is associated with the catalytic activity, and that can be identified as a portion which deals with the inhibition activities. The active site occupies only a small portion of the whole enzyme molecule, and its configuration is determined mainly by configuration of the whole molecule. Binding of a substrate with the active site of an enzyme occurs through a non-covalent bonds. These forces are hydrophobic in nature because the environment of the active site is hydrophobic. Water is admitted only when it is a reactant. Usually an active site is formed of 12 to 16 or more amino acid residues known as active group, catalytic groups, catalytic residues or functional groups. These are located at distant points in the polypeptide chain, often at opposite ends. The specific, usually the active site is formed of 12 to 16 or more amino acid residues known as active groups, catalytic residues or functional groups. These are located at distant points in the polypeptide chain, often at opposite ends. The specific folding of the polypeptide chain brings them to close proximity. Usually, the active site is formed of 10, 12 to 16 or more amino acid residues known as active groups, catalytic residues, or functional groups. These are located at distant points in the polypeptide chain, often at opposite ends. The specific folding of the polypeptide chain brings them to a close proximity. The remaining amino acid residues of the chain determine the molecular form of the enzyme and influence the reactivity of the functional groups. Those amino acids seen close to the active site are capable of influencing the reactivity of the functional groups are called accessory groups. Those amino acids seen close to the active site and capable of influencing the reactivity of the functional groups are called accessory groups. Remote amino acid residues capable of influencing the molecular configuration of the enzyme are called facilitating groups. Once again, those amino acids seen close to the active site are capable of influencing the reactivity of functional groups are called accessory groups. Remote amino acid residues capable of influencing the molecular configuration of the enzyme is called facilitating groups. So accessory groups will be there and facilitating groups will be there. The binding of the substrate to the active site depends on the proper alignment of the functional groups. Since the active site has no rigid and constant shape, the functional groups align themselves to promote substrate binding. So unfolded peptide chain is there, functional groups are there, and functional groups are folded peptide chains are also happening. The binding of the substrate to the active site depends on the proper alignment of the functional group. Since the active site has no rigid and constant shape, the functional group realign themselves to promote substrate binding. So now let's learn about the allosteric enzymes. Allosteric enzymes, an important topic. In some conjugated enzymes, one or more regulated or allosteric sites may also be present, distinctly separated away from the active site. Allo means other. Such enzymes are called allosteric enzymes. In configuration, the allosteric site is somewhat similar to the active site, but it has no catalytic site. It has only contact site. The molecules capable of binding with allosteric sites are called allosteric regulators, allosteric effectors, or allosteric modulators. They are sterically or structurally different from the substrate. Though binding with the allosteric center, they exert some influence on the binding. Though binding in the allosteric center, they exert some influence on the binding and conversion of the substrate at the active site. Their effects may be excitatory or inhibitory. So, allosteric effectors can be positive regulators or negative regulators. Positive regulators are generally called activators, and the negative regulators are called inhibitors. Some allosteric centers are specific to activators and others to inhibitors. The action of allosteric inhibitors on the enzyme occurs through a conformational change in the active site. On the other hand, allosteric activators promote the catalytic conversion of the substrate by increasing the velocity of the reaction. Enthalpy, entropy, and free energy. Enthalpy, which is represented by H, and entropy, S, and Gibbs free energy are three thermodynamic quantities which explain the energy changes in living systems. The changes of these quantities are represented respectively by delta H, delta S, and delta G. Enthalpy, entropy, and Gibbs free energy. The changes of these quantities are represented respectively by delta H, delta S, and delta G. In the standard state of biochemical systems, they are written as delta H0, delta S0, and delta G standard. The standard state of biochemical systems is the condition of which pH 7, temperature is 25 degrees Celsius, solute concentration is 1 molar, and the pressure of all gases is 1 atm. So that is the standard condition. First, let's learn about what is enthalpy. Enthalpy is the total heat content of a system. It represents the number and kinds of chemical bonds in the reactants and products of the reaction system. It is expressed by the equation H is equal to capital E plus PV. H is equal to E plus PV. H is equal to E plus PV. Pressure, volume, work, and where E is the internal energy and P is the product of pressure and volume. H is equal to E plus PV. Okay, where E is equal to internal energy and PV is the product of pressure and volume. The reaction system that takes up heat from the surrounding is called endothermic system, and it has positive value of delta H. On the other hand, the reaction system which releases heat to the outside is called exothermic system. Heat releasing type is called exothermic system. Releasing. Okay. And it has a negative value of delta H. Delta H is negative in case of exothermic system, which releases energy into the environment. Next is the entropy. Entropy is the degree of energetic randomness of a system. It is a measure of the energy that is not accessible or available for useful work. It's a degree of randomness, but that randomness is of no use because that can be converted into available or useful work. Entropy and enthalpy together quantify the third quality, namely free energy. Concept of free energy. Gibbs free energy is a part of a total energy system available for work during a reaction under conditions of constant temperature, pressure, and volume. The portions which are not immediately available for work is called bound energy. The difference between the free energy content of a reactant or substrate and the free energy content of its products is called change in free energy level or standard free energy level or delta G. It represents the maximum amount of energy made available during the conversion of a substrate to a product. When a reaction proceeds with the loss or release of a free energy, the free energy content of a product is lesser than that of the substrate, and delta G, free energy change, will have negative value, negative delta G. In such cases, the reaction will be exergonic. Earlier, we have learned a term called endothermic. Endothermic, in the sense heat is absorbed by the system. Endothermic reaction where delta H was positive. And the other term is exothermic. Exothermic in the sense heat is released into the environment. So the delta H is negative. Here we are learning another term which is exergonic process. Exergonic process. Well, When a reaction proceeds with the loss or release of free energy, the free energy content of the product is lesser than that of the substrate, and delta G, free energy change, will have negative value delta G. In such cases, the reaction will be exergonic. Exergonic energy will be released. There will be decrease in free energy level, and G will be available to do work or to be released as heat or for both. On the other hand, when a reaction proceeds with gain of the free energy content of the product will be higher than that of the substrate. The delta G will be positive. In such cases, the reaction will be endergonic. When delta G is positive, the reaction is endergonic. There will be energy absorption and increase in free energy level, and no G will be available for work. So when the process is endergonic, no delta G, G is available for work. So such reactions occur only when there is an input of energy to drive them. Anabolic reactions are all endergonic, and catabolic reactions are exergonic. Catabolic in the sense breaking reactions. Changes in free energy are always related to changes in heat and entropy. The relation between the changes in free energy, heat and entropy can be represented by the equation delta G is equal to delta H minus T delta S. Delta G is equal to delta H minus T delta S. Delta G is equal to delta H minus T delta S. Okay, so that's the major equation. In this equation, H is the heat produced or consumed, T is the absolute temperature, and S is the entropy. This equation very well reveals that in a reaction which produces no change in temperature, there will be a decline in free energy. If there is rise in entropy, energy always flows in the direction of increasing entropy. Oh, that's a new point. Energy always flows in the direction of increasing entropy. So chemical reactions can proceed simultaneously and spontaneously if they produce an increase in entropy or a decrease in free energy. Thus, free energy appears to be the driving force in enzymatic reactions. So lowering of the free energy of activation. Activation energy or free energy of activation is the energy required by a reaction to make a substrate reactive for its reaction conversion to a product. In effect, it is an energy barrier for the reaction, high in non-catalyzed reactions and low in enzyme catalyzed reactions. In effect, it is an energy barrier for the reaction, high in non-catalyzed reactions and low in enzyme catalyzed reactions. The level of the free energy of activation is an index of the rate of the reaction, and both of them have an inverse relation to each other. So higher the activation energy, the lower the rate of reaction. Conversely, the lower the energy of activation, higher the rate of reaction. Enzyme react function as catalysts and thereby lower the level of the free energy of activation, reduce the energy barrier, and enhance the overall reaction rate without altering the temperature level. In brief, enzyme increase the rate of reaction by lowering the activation energy. This can be compared to crossing over to the other side of a mountain through a tunnel instead of scaling through the peak and when climbing down to the valley of the other side. The greater the amount of activation energy required by a reaction, the slower will be the rate of reaction at a given temperature. The free energy required to lower the level of activation energy is called intrinsic binding energy. The free energy required to lower the level of activation energy is called intrinsic binding energy. It is derived from weak non-covalent interactions. That is a new term. Intrinsic binding energy. The energy required to lower the level of activation energy is called intrinsic binding energy. It is derived from weak non-covalent interactions between the enzyme and the substrate soon after enzyme substrate complexing. Each weak interaction is accompanied by the release of some free energy. This energy is called binding energy. The free energy required to lower the level of activation energy is called intrinsic binding energy. The free energy required to lower the level of activation energy is called intrinsic binding energy. It is derived from weak non-covalent interactions between the enzyme and the substrate soon after enzyme-substrate complex. Each weak interaction is accompanied by the release of some free energy. This energy is called binding energy. It stabilizes the enzyme-substrate interaction and also lowers the level of the activation energy of the reaction. The amount of the activation energy lowered by an enzyme is called catalytic efficiency. Catalytic efficiency. Intrinsic binding energy and catalytic efficiency. Delta G is equal to change in free energy. G is equal to free energy. Delta Gc is equal to activation energy for enzyme catalyzed reaction. Delta Guc is equal to activation energy for uncatalyzed reaction. Delta G is equal to catalytic efficiency, the amount of activation energy lowered by enzyme. Delta Gb is equal to intrinsic binding energy. What is that intrinsic binding energy? The free energy required to lower the level of activation energy. Lower the level of activation energy. Okay, so delta Gb is equal to the amount by which the activation energy is lowered. This means that Gb is equal to delta G delta B, catalytic efficiency. This stabilizes the enzyme substrate interaction and also lowers the level of the activation energy for the reaction. The amount of the activation energy lowered by an enzyme is called catalytic efficiency. The amount of activation energy lowered by an enzyme is called an catalytic efficiency. The amount of free energy required by an enzyme to lower the amount of catalytic efficiency is called intrinsic binding energy. That is the amount of energy required to lower the level of activation energy. Mechanism of enzyme action. Let's learn about the mechanism of enzyme action. Enzymes play a pivotal role in virtually all metabolic reactions. Different enzymes catalyze their respective reactions with different efficiency. Their catalytic efficiency depends mostly upon the way in which they react with their substrate. The actual mechanism of enzyme action is not fully understood. In fact, enzymes only promote biochemical reactions, which can proceed even in the absence of enzymes. So, enzymes never influence the direction of reactions, but only enhance their rate of velocity. They increase the velocity, but they don't influence the direction of reaction. Theories of enzyme action. An overall theory of enzyme action has not been advanced so far. Still, it is generally held that enzyme action, in all probability, involves three aspects, namely enzyme-induced lowering of the activation energy, high specificity of enzyme action, and precise regulation of catalytic process under biological conditions. Precise regulation of catalytic process under biological conditions. At the beginning of the last century, Bayliss and Warburg advanced the adsorption hypothesis to explain the mechanism of enzyme action. According to this hypothesis, the surface of an enzyme provides suitable sites for the absorption of reactant molecules. This adsorption facilitates enzyme substrate interaction to accelerate the reaction site. At present, this hypothesis is only of historical importance. Next, the enzyme substrate complexing hypothesis will be there. That is the Michaelis-Menten theory. A significant advancement in the understanding of enzyme action was the formulation of the enzyme substrate complexing hypothesis by Leonard Michaelis and Mendel, which we just mention in the next video, and we will be explaining it in a more detailed manner. Thank you for listening.